Excellent. Well, good evening, everyone. Thanks so much for coming and braving the truly terrible Hobart weather out there. It's nice to see some faces. I'm actually really pleased to actually have this in person and online as well, because obviously there was some doubt that it could happen, but I'm very pleased that it's going ahead as planned. So great to see you here and online. So today we're meeting on Lutruwita, Tasmanian Aboriginal land, sea and waterways. I acknowledge with deep respect the traditional owners of this land, the Moonina people, which we meet on today. Our island is deeply unique with spectacular landscapes, with our cities and towns surrounded by bushland, wilderness, mountain ranges and beaches. I stand for a future that profoundly respects and acknowledges Aboriginal perspective, culture, language and history, and the continued effort to fight for Aboriginal justice and rights, paving the way for a stronger future. So my name is Nick Fountain-Jones, Dr. Nick Fountain-Jones. I am an infectious disease ecologist, so as you can imagine, I've been pretty busy of late. I would have imagined when I first changed disciplines to become an infectious disease researcher that I would ever have the terms genomic sequencing or super spreading as the modern vernacular. But this is the world we live in. I currently do my research in maths and physics in the School of Natural Sciences at UTAS, so it's a good place to work and be, and model infectious disease. The idea behind this symposium was to bring people, experts from around Tasmania and abroad, thanks Erik, to actually give some perspective, give some idea of what we're going to live through collectively over the next couple of months, couple of years. Okay, so of course, we all are very painfully aware that the world has turned upside down with COVID-19. But it's always good to keep in perspective that what we see now is a result of one event, one spillover from a wild species probably into humans is responsible for all of this. And these spillover events are becoming increasingly common. Of course, this happened in China around Wuhan in 2019. And these events are just going to be increasingly common in the future. And I think this highlights the need for understanding how wildlife health, human health, and domestic animal health and environmental health all come together to predict spillover. And that's why I think I was excited to help organise this tonight. Okay, and I think this research is crucial for actually predicting and stopping the next pandemic. So that's a bit about my background at least. Of course, this has involved, since the pandemic has started, it's become a huge scientific achievement getting as far as we've got with vaccines and all the data analysis and modelling which we have at the moment. It's a huge scientific achievement involving lots of different disciplines, from molecular virologists through to social scientists, and all offer very important insight into the pandemic. And in our panel, our wonderful panel, we have experts from some of those disciplines here today, which is very exciting. Before we get going, can I just ask a question for audience? Who's vaccinated? Who's had their... Nearly everyone? Who's had at least one dose? Yep, 2 doses. Maybe got the wrong audience, but anyway, that's all right. Vaccines are, of course, a key part of this and the race to herd immunity. But as you find out tonight, herd immunity might be a mirage, of course. So it's not plain sailing from here at all. But without further ado, I'll introduce our fantastic set of panellists, which are going to talk all about this topic. And we're really excited to have this fantastic group of world experts right here in the room and on Zoom. So we've got Erik Volz, Dr. Erik Volz from Imperial College London. How are you doing, Erik? Can you hear me okay?
I hear you great, Nick. Thank you.
Excellent. And Erik has been a really important cog, so no pun intended, in the UK genomics effort, I suppose. And he's been particularly important in understanding variants of concern and identifying them from data sets. So it's really great to have Erik with us. We've also got the fantastic Mike Charleston over here. His expertise is in mathematics and infectious disease and bioinformatics. And Mike's going to talk to us today about how good models are and what we can use them for. So it's really great to have Mike. Then we've got team immunology. We have Andy Flees over there Dr. Andy Flees from Menzies. And we have Associate Professor Guna Kurupiah over there as well from School of Medicine. And these guys are going to talk about immunology and vaccines, which of course is a central topic. Then we have the fantastic Associate Professor Nicola Stephens over there, and she is also in the School of Medicine, but she's also the strategic leader for epidemiology in the, let me get this right, the Tasmanian Public Health Emergency Operations Centre, which is a fantastic centre managing our COVID response. So it's really great having Nicola here with us. So that's our fantastic panel. I'd just like to give a warm welcome to them. So can everyone give a round of applause? This fantastic panel. I'm really excited this has come together. So without further ado, I'll hand over to Erik Volz, and he's going to talk to us about some genomic sequencing stuff he's been working on. So over to you, Erik.
Thank you, Nick. Give me a moment to share my screen. And please let me know if we have any sound or audio problems. Do you have my screen?
Yep, we do. Fantastic.
Okay, wonderful. It's nice when video conferencing technology actually works, especially when you're making a call all the way across the globe. So thank you, Nick, for the introduction. Thank you to the organizers for inviting me to this wonderful seminar. And good morning from London. I know that we all want to get some insight this evening or morning into the next few years of the COVID-19 pandemic. I want to start by tempering expectations about that. It's in my view, the future is very, very murky. The problem is that the future course of the pandemic depends on many, many variables. It's in some ways more complex now than it ever has been. So I don't think anyone can make any very solid predictions about how the next couple of years will play out, but we can at least say what is likely to happen if certain variables change. So to boil this down as much as possible, I think the future of the pandemic depends on our behavior, so what people do. It depends on what our immune systems do, and it depends on what the virus does. Different features have been important at different phases of the pandemic. So in 2020, I think behavior was overwhelmingly the important thing, determining what the pandemic did, especially public health interventions. But now we have this very complex landscape of immunity, so various levels of vaccination or natural infection, and things that we are still uncertain about. So for example, how quickly immunity wanes. How quickly does immunity wane after natural infection versus vaccination? What type of vaccine you have? What if you have been vaccinated and then acquired natural infection? So the landscape of immunity is extremely complex, and we're only just beginning to be able to measure those sorts of variables. In this talk, I'm not an immunologist at all. I'm a genomic epidemiologist. I'm going to talk about what the virus has done. So this will be a bit of history about how the virus has changed over the past year and a half. And I'll talk a little bit about how we actually detect those changes, which will be important going forward. So I think most people in the audience will be familiar with the main variants of SARS coronavirus 2 or SARS-CoV-2. We know them as alpha, beta, and delta, and so on. And now I think most people know that delta is the predominant variant the world over. But the story of SARS-CoV-2 evolution predates that. We've actually seen major changes in the virus occur on at least 3 separate occasions, and the first occasion was quite early on. In January of 2020, a mutation was detected on the spike protein. This mutation was called B614G, meaning it was a change from an aspartic acid to a glycine at position 614 of the spike protein. And in the subsequent months, many scientists noticed that the clade of the virus, so that is the group of virus lineages that are closely related, which contain this mutation, seemed to be spreading more quickly and more widely, or over a much greater area. And the question was, was this a coincidence, or was it representing a change in virus fitness? This, one of the first scientists to note this was actually our previous speaker, Nick Fountain-Jones. This is actually how we became acquainted. In the spring of 2020, he used one of the methods that we developed in order to detect a significant difference in the clade containing this variant, and that's highlighted in red. The important thing to note about this clade is that it began expanding very, very rapidly in the spring, and its origins could be traced to the founding of the epidemic in Europe. And of course, in spring 2020, the epidemic began growing very, very fast in Europe and North America. So the question was this mutation hitchhiking along the success of the virus in Europe and North America, or was the severity of the epidemic in those places in some way a consequence of this mutation? So another very early study into this mutation was now famous study by Betty Korber, who noted that the rate that the frequency of this mutation, 614G, was increasing was consistent with a selective advantage. And she further did some lab studies showing, for example, higher rates of cell infectivity and higher viral loads in patients infected with the variant with 614G. So this was all sort of bolstering the case for there being no selective advantage. But surprisingly, in summer of 2020, the attitude of most scientists and experts in this field was skepticism. Everyone acknowledged that after entering a new host, a virus is likely to change and make some adaptations to a host. But we actually have fairly few historic precedents for a major change in transmissibility. We know it can happen, but there aren't many examples of it happening and where it being really well established. So the attitude of most scientists was skepticism. We knew the virus would adapt, but we didn't think it would necessarily make huge changes to transmissibility. So that's where we came in the UK. We leveraged a wonderful data set compiled by the COVID-19 Genomics Consortium, or COG UK for short. And in the summer of 2020, this was by far the largest data set of SARS-CoV-2 genomes in the world at a national level, and also the highest coverage, meaning we were sequencing about 10% of diagnosed cases. And we've actually maintained that level of coverage up to the present. So with the advantage of having this very deep data set, we could do phylogenetic analyses and really pin down the likely size of the selective advantage of 614G. The way we did this was to reconstruct phylogenies, which included sequences from both the UK and worldwide. We would reconstruct the ancestral states. So we would say if a lineage was likely within the UK or outside the UK. And then we would pull out clusters of lineages representing transmission within the UK. So once we have these clusters, these are like independent data points representing outbreaks. And then we can compare outbreaks with the 614G variant versus 614, the ancestral 614D variant. And we can see if, for example, the outbreaks with G tend to be larger and grow faster. So that's what we did in this paper published earlier this year. And we showed, like Betty Korber did, that the rate of growth of the 614G variant was consistent with the selective advantage that's shown in the bottom right panel. But we could also show that the size of outbreaks was larger with the 614G variant. That's what's shown in the top right panel. And doing a bit of mathematical modeling, we could also estimate the size of this effect. So in the end, we estimated that although it was still very uncertain, we think the transmissibility change was about 20% with this variant. And at the time, we thought 20% was actually quite large. And the these findings were later bolstered by both other mathematical modeling studies as well as lab studies. So I think the most convincing results were actually carried out by Ralph Barrick's group, where they directly estimated transmission difference in animal models and hamsters, and they could actually directly measure that the 614G variant was being transmitted more quickly. So, so this is all a bit of ancient history. I mean, we thought 20% was a big transmissibility difference, but we had no idea what would happen several months later. And to me, this was actually one of the most surprising things of this pandemic. We've seen subsequent huge step changes in transmissibility with the Alpha variant and with the Delta variant. And now I think if you read the newspapers, almost weekly, there's a story about a new variant. Usually these aren't very important changes, but it's very much at the top of the public consciousness now. So how did we go a year ago from a situation where the public was mostly unaware of this sort of situation, and most scientists were skeptical, to a situation now where there is weekly newspaper stories and weekly science studies about the latest scary variant. So here's a bit of history and an anecdote relating one of, well, this is actually the most interesting episode of my work in the pandemic to date. This is about the origins of the alpha lineage, which we call B117. So the story begins in November 2020, when on a WHO call, a scientist inquired about a particular mutation, N501Y, and they were worried about it in their data set in South Africa because there had been a study, a lab study in humanized mice showing that this mutation developed very quickly in mice and was on the receptor binding domain, so it could plausibly influence infectivity and antigenicity. So we had to look at our own data set just to see if this mutation was present or not. And when we did that, the results were quite startling. We saw that not only was the mutation present, it was in an unusual clade of the virus and was growing very quickly. So I show these figures. These are not particularly good looking figures, but I show them for historical reasons, because this is actually the first phylogeny estimated of the alpha lineage and the first plot showing growth of that lineage. We made this in early December, and two things to note about this phylogeny is the very long branch leading to the B117 clade. That means that there were a large number of genetic changes in this lineage relative to its nearest relatives. And another interesting thing about this was the presence of a particular deletion in the spike protein. So that is to say, 2 amino acids were actually dropped from position 69 and 70 of the spike protein. The significance of that is that it influenced diagnostics. So that actually caused a common diagnostic for COVID infection to fail, a so-called Takehome assay. And so there was some concern that we might be underdiagnosing this lineage. We now know that we probably weren't underdiagnosing it to any great extent, but this actually turned out to be a weird bit of good luck. The reason this was good luck is that the Takehome assay tested for several genes in SARS-CoV-2. This deletion caused failure to detect the spike protein, but it didn't cause failure to detect the other proteins. So you could still get a positive COVID diagnosis, but if you failed to detect the spike protein, that was a very sensitive and specific indicator that you were sampling a patient that had this lineage. So this was actually a proxy way of detecting infections with this lineage, and that greatly expanded the amount of data we had to work with. This plot on the right shows the frequency of the N501Y mutation over time. This also the first figure that was made showing this. This is in log odds terms. And so when you see that linear trend in log odds terms, that basically means the frequency is growing exponentially. And doing a little bit of modeling, we can translate that growth rate into the likely transmissibility advantage. And it came out as being 70% per generation. So that is to say that this might indicate that someone infected with B117 lineage would infect 70% more people than someone infected with the ancestral type. So this was all a bit concerning, but we were still a bit cautious at the time because we didn't want to raise a false alarm. But unbeknownst to us at the very beginning, there was a parallel investigation by Public Health England who had noted a very large and fast-growing outbreak in the southeast. So in November of 2020, the UK was actually in lockdown. Many businesses were shut, but schools remained open. And so schools remained one of the few ways that you could get self-sustaining transmission chains. And the only place in the UK that had growing incidence was Kent in the southeast. In early December 2020, we put our heads together, the genomic epidemiologists and the health protection teams. And we put these pieces together, and we noted that when you look at all local authorities in England and compare the prevalence of B117 against weekly incidence, there is a fairly strong linear correlation. Again, this is actually the first figure that we actually passed up to the Chief Medical Officer's office. And so by this point, when we put these pieces together, the level of concern jumped very, very high, because now we had sort of a mechanism and a lot of circumstantial evidence that this was driving growth, and probably by quite a large extent. And if you use models to predict how quickly this variant would spread, the predictions, which turned out to be true, were very concerning. So we predicted that it would be more than 90% of virus by the end of December, coinciding with the time when lockdown was to be lifted. So it is potentially an emergency. And at this point, yes, go ahead.
Hi, Erik. Just letting you know, we've got 1 minute before we need to wrap up. So for the next speaker, is that all right? So
yeah, absolutely.
Thank you.
So at this point, dozens of people get involved, and actually, I think they moved very, very fast. Less than 2 weeks later, a second lockdown had been elapsed. I'm somewhat proud of this moment where Boris Johnson quotes two numbers from produced by our research group. This was actually a historic event, because I think this is the first time that genomic epidemiology had been used to detect a variant of a currently circulating pathogen, which actually drove a public health intervention over a very, very short time. So this shows very nicely, I think, the promise of genomic epidemiology, particularly when you have these very large, well-curated data sets. So with the second lockdown at the beginning of 2021, lockdowns really work. There was some doubt, given the higher transmissibility, that this would work, but actually cases began to decrease almost immediately, and would continue to decrease until the Delta variant came along. And where we are today is we have a great many more people, and we have a greater sophistication and methodology to detect these variants in the future. And the shift is very much more on antigenic changes now, because there are very high levels of immunity in the population. At least in the UK, not necessarily in Asia Pacific region, any successful variant will likely have to feature some major antigenic changes. And so our surveillance systems are now very much geared towards detecting those. Thank you, Nick. I think I just managed to finish on time.
That was a really great update to the new stuff too. I haven't seen that. That's amazing. Thank you so much, Erik, for that talk. What we'll do, how we'll run this is we'll just give the presenters their time and then I'll move on to the next one. But make sure you write down your questions and we can ask questions at the end. So hold off on the questions. And I'll introduce Professor Michael Charleston. Over to you, Mike.
Okay, cool. Thank you. Thank you, Nick. Right. Can you hear me? Yes. Good. I don't need that. Does this work? Don't know. Right. Okay. That one? Okay, cool. All right. So I'm a mathematician. Nick and I work together on this kind of stuff. Mainly concerned with wildlife epidemiology, but so I haven't actually been doing any modelling of SARS-CoV-19. Don't take what I say as being this is what's happening, because I'm not actually doing the modelling for it, but I do know roughly how you do some modelling. And so I just thought I'd share with you some of the things that models can tell us, what a model is and what it can't tell us. And I'm going to lead with, when this is going to end, because as we've just heard, there are just so many unknowns, so many difficulties, and so many things we cannot predict, simply cannot predict, not just not know about now, like what's going to happen evolutionarily, that we can't predict when things are going to just change massively. So that's one thing that we can't do. But I guess one thing that we can do is understand a little bit more perhaps what a model is. And so I thought I would just share some thoughts with you on that. There are three basic kinds of models. We can talk about physical, concrete models, like your favorite ethics construction, which I didn't make that one, it's far too good. And that's a physical model or a concrete one. Another kind of a model that we might think about is a conceptual model. The one on the right there is an idea of how the carbon atom works, what it looks like. We've got electrons floating around the outside. We know physically it doesn't really do this, but it helps a lot in our understanding. The big one there that's relevant to us right now is a mathematical or a computational model, which is about how disease spreads in the population. So that's a very standard model known as the SIR model, where you have a proportion of the population that is susceptible, proportions infectious, and a proportion that is recovered or removed from the population. And we use a set of equations to describe the rates of the population moving between those three different conditions. So a little bit more about that in a minute, that particular model. Now, I will say that models, you'll probably have seen a lot of models being talked about, and you would have remembered models from climate science. People talk about running models. Models don't have to be right to be useful. There are an awful lot of ways of modeling epidemiological spread, of modeling climate, modeling temperature, or modeling economics. It's great if we don't have to get the model exactly right to get the right answer, and in general that is the case. So there's a famous phrase, all models are wrong, but some are useful. We hope that these are useful models. For a model to be useful, it has to be testable. It has to make predictions that we can then verify with observations or with data. We hope our models are useful in that sense. And of course, we're today very interested in epidemiological models, and these are going to make predictions about how disease spreads through a population. Here is that very, very simple model, the SIR model I mentioned. It has three different categories of being in susceptible, infected, or removed. Removed could be recovered. Removed could be removed from the population because you've died. So that's how that set of equations works, differential equations. And differential equations, sets of DEs like this can be solved numerically or analytically. So you get an exact answer in that case. Mostly DEs that we use to model epidemics are more complicated than that and have to be solved numerically. But you can see that there's this characteristic curve that you may be familiar with already. The red one on that bottom plot is actually, does this work? Is this a pointer? No. Yes? That's a clicker. I don't want to do that. Okay, I'll just point over there. There's a red one. Also, I can use, colors. Cool. It's very bright. Yes, it is. So we've got this beautiful characteristic Beautiful. Well, it's a characteristic curve in red there. Sudden rapid rise, a peak, and then a slower decay. And that we have seen many, many times. And that's the number of infected in the population. And the other two curves there are the number initially susceptible, and the black line there, the recovered or removed from the population. So we can solve these equations, see those curves in real situations, and there's one there. It looks vaguely familiar. It sort of fits that characteristic. And these are cases in Australia up to a couple of days ago, and you can see we haven't peaked yet. So yeah, we're not out of it. Models can be deterministic, like those differential equation models, or stochastic. Stochastic really means having some randomness, some effective chance in them. And I'm a big fan of stochastic models because I don't then have to solve systems of differential equations to work with them. And they capture a couple of great things that differential equation deterministic models do not, and that is randomness, of course, and variability. And they can be made much more structural. You can construct a model that has structure in, say, the population, like putting people in towns. Are you looking at me to hurry up, or because you're paying attention? I'm doing my best, okay. So here's a quick one, which is modeling the spread of infectious disease on a population, and I'm really glad that that is looping correctly. And that's a population of just 100 nodes, and it's a really, really simple model. Everyone can meet everyone else, and they can spread an infection, and it starts with just one infection, and then it rapidly spreads through because everyone can communicate with everyone else. There's no social distancing, everyone's traveling everywhere, disease spreads rapidly all the way through the population, and for moments there, everyone's infected. Right, it's very bad, okay? We should, in fact, what we should do instead is split up our population into something like suburbs and stick to our suburbs. And this is the same parameters of that simple model. I won't go into the details of the code of it because it's not here. But under that model, is it going to start again? I think I have to go back again. Go back. It starts and then it's over, right? Same parameters. There's not nearly as many people to meet. Disease stops. So we can model structure into our put structure into our model that way, and we can include a level of idiocy, which is always entertaining. And you can see what happens. Now that's just exactly the same parameters once more. I'm going to go back and show you again because is that the first? Yep. Okay. You see what happens. The idiots are the people who travel between suburbs. All right, so if you stay in your suburb, you're not an idiot. If you are an idiot, then you can travel anywhere, and you can see that's a terrible outcome. So that's one of the things that differential equations can't really take into account. That's one of the things that modeling can sort of allow for and show, but they can't tell you when there's going to be an idiot, and that's obviously a problem. So I set up another little model, and I'm just going to go straight to the link, and I really hope it works because I haven't tried it out. Let's see. And this is taking, oh God, do I have to do a thing? It's probably a virus, yeah, thank you. All right, so yes, that seems to be working, which is awesome. All right, so I don't know if you can see on the screen there. Might need to zoom in a bit. All right, let's see. Can you see the numbers on there? Yes, you can. I hope so. So we've got a population size of 200. We've got a maximum probability of moving through this population. We have just one suburb, and with any luck, we're just going to be able to see some of the different variation that you can expect. And here's one. So I've got 11 simulations of the same set of parameters. And you see there's quite a lot of variation there. So this is why modeling populations is great, but we can't predict what or when things are going to happen. I can show you one more thing, though, which is the effect of splitting people into suburbs without idiots. Again, so that shows a huge drop in that blue line is the average number of infections per day. All right, so it drops right down. Let's go back again to 0 or one village. It's a heck of a lot more, right? So if you split people up, great. And all of the models tell you. Another point, The simulation that I did before, differential equations, they will all tell you the same kind of thing. So the models might be wrong, but they're giving us the same answer. And now let's add in a sort of quota of idiots. Let's bung in, say, that many, and you can see that it does make things substantially worse again. And it also seems to, often I have found when I'm running these things, it seems to make things quite less predictable as well. There's a lot more variation, and that makes sense because you never know when people are going to jump between a suburb and have another outbreak. So that was quite fun to play with, and I hope, and this is an app that you can actually have a look at, play with yourself as well, and tweak those parameters. Is it that one? Yes. And I think that's actually all I wanted to say. So just to give you a little bit of an overview on what models can and can't tell us, and that's all. So thanks.
Thanks for that talk, Mike. A really fun app. Feel free to download and use yourselves. Really fun. I was on the web, sorry, my apologies. Next, we've got Team Immunology, leaded up by Andy, please. So, go away, Andy.
All right, thank you, Nick. I'm going to see if I can use the pointer from this. Might be easier. We'll see if we can get by there. All right. So thank you for organizing this. I'll try to stay behind here so the microphone will pick me up properly. And it was nice to have that. And those first two have talked about how we track the changes to the virus and then how we understand where the virus is and where it might be going. So I'll focus on vaccines today with the idea of I'll show you a little bit of what's under the hood of how to make a vaccine and show you and hopefully give you a better idea of the process and the timelines that new vaccines could potentially be made. So this is not showing up quite as I hoped it would, but there's a lot of different ways to make vaccines. There's probably 10 different common ways to do it. And the simplest one is this one, where you take the pathogen, which is the virus. In this case, this is SARS-CoV-2. You grow a lot of it in the lab, and then you kill it through chemical inactivation or some other form. Then you can put that back into a person, they'll develop immune response against it. It's probably not going to be as strong as putting in the live virus because it's just a dead virus. It's not doing anything, but you can get a pretty good response that way. And there's one that's been quite widely used, and it, for the most part, has worked pretty well, but recent studies are suggesting it's maybe not working as well. Then we have these other two down here, and the top one is viral vectored, and the other one is a nucleic acid vaccine. So the nucleic acid vaccine, that's the mRNA vaccines that most of you are probably hoping you would get or waiting for. It's the Pfizer one and the Moderna ones. And the viral vectored ones are mostly based on using an adenovirus and dressing that up to make it look like a different virus. So whenever I talk about these through the next few slides, I'll show this for the adenovirus vaccine and this for the mRNA vaccine. So there's a lot of vaccines being used around the world right now. This is pretty unprecedented to roll out this amount of vaccines in this short of time. The AstraZeneca is actually the most widely used vaccine. It's used in 184 different countries. A lot of that is because they have most of the African countries where the logistics and distribution may not have the cold chains that allow the Pfizer to be distributed, and it's a little bit easier to produce that one. So Pfizer is obviously very widely used. Interestingly, this Gamaleya one from Russia, it appears to be a very good vaccine, and they did something very clever right on. So they used two different viruses. So you get the initial priming dose with one type of virus, and then the second one is a little bit different. Because when your immune system sees that first virus, it makes an immune response not only against the target, the coronavirus proteins, but also against the viral vector you were using, the adenovirus. So if you use a little bit different adenovirus for the second one, it slips past your immune system, and then it can target the coronavirus proteins instead of the adenovirus. So that one works pretty well, but Russia's not forthcoming with all of their data, so people still don't quite trust it. All right, so the mRNA vaccines, I'm just going to do a quick coverage of this because it's not something that I really have expertise in. People have been working on this for a long time, but it hasn't been widely used in the public until this year. So these are the Pfizer ones, the Moderna, and then there's another one from CureVac, a German company. And with the Pfizer and Moderna working so well, I think everybody thought, wow, we have this new type of vaccine, it's going to solve all our problems. But the CureVac, the latest results just came in, and it was only about 50% protection compared to the 90% protection from the other two. And the company claims that it's because when they gave the high dose in the early clinical trials, it was not as tolerable. People really didn't like getting it, and you can't have that, those bad adverse events. So they dropped the dose down, and then the response wasn't that good. So with any vaccine and with any type of medicine, the dose will make the poison. You want as high as dose that's tolerable to the patient without causing adverse events and gets the job done. What's really exciting about these mRNA vaccines is they can be used for a lot of other things now. So they're already being rolled into cancer immunotherapy trials with some of the other effective cancer treatments. And they're being developed for other viruses. So for influenza and cytomegalovirus, a really common virus in the human population, and also for things such as cystic fibrosis. So we'll really look forward to seeing where this type of vaccine goes in the next few years. And now I'll switch over to the adenoviral vaccines, because that's what my team at the Menzies Institute for Medical Research is actually making. We're not making one for the coronavirus. We're making it to try to help Tasmanian devils. But we're using the same viral vector that's being used in the Russian vaccine and one of the Chinese vaccines. So how do you make a virus look like a different virus? The most important thing to have right away is the genetic code, and we get that from people like Nick and Erik that are sequencing the genomes. And once we have those genetic codes, we can start manipulating it and make the virus into a vaccine. So most of the vaccines have focused on this spike protein on the outside of the coronavirus. So this is how it contacts cells and enters cells. So if you can make an immune response that blocks that interaction with the host cell, you can block infection and the virus can't make copies of itself. So the way to make this into a vaccine is you take that genetic code that codes for that spike protein, and I apologize for that getting cut off a little bit there, but then you splice it into the genome from the adenovirus. So an adenovirus has about 36,000 nucleic acids in it. The spike protein, if I remember correctly, is about 1,800, the adenovirus has about 36,000 nucleic acids. The spike protein is a little less than around 2,000 nucleic acids. So you cut that DNA out, you put it together with DNA for the adenovirus. And the really important thing here is that when you put that DNA from the spike protein in, you cut out part of the adenovirus. And that part we're cutting out here is the part that allows the adenovirus to make copies of itself. So you've just attenuated that adenovirus by taking out a key part, and instead it's got that coronavirus DNA in there. When the virus is then put into a cell, it'll try to make a copy of itself. It can't, but in the process, it makes the spike protein. Your immune system will recognize the adenovirus because your immune system is very good at picking up on when virus is there, so it attacks the adenovirus or what's there of the adenovirus, and at the same time, it picks up the protein from the coronavirus too. So how do we, once we've spliced that genetic code together, how do we turn that into the virus? So I did that for the first time this year. Actually, our research assistant, Jocelyn Darby, did, but I did the design on it, so I'm going to take somewhat credit for it. But it's a pretty mind-blowing process the first time I did this. So we walked into a room, we had the genetic code for the adenovirus with some of our devil facial tumor proteins in it, and we mixed it with some cells, and then we put it into this machine over here called the nucleofector 4D, and it zaps the cell with a bit of electric current. It carries the DNA inside the cell, and it starts making the virus. And what's really critical about this is this cell that we were using is called a helper cell line. So it has the part of the adenovirus that was missing from the genome. So it makes that protein and allows the adenovirus to assemble into a virus. It's like a bunch of Legos, and you have all the parts only when it's in that cell. So when it goes in somewhere else, it's missing one of the parts, and it can't assemble the new virus. Once you do that, you purify the viruses, and then you find a willing participant who does not have that missing part of the adenovirus. Hopefully, the virus tries to copy itself. It can't. It makes a bunch of viral proteins, and it'll make that SARS-CoV-2 protein in the process. Your immune system comes in, clears out the adenovirus, and also attacks the coronavirus protein at the same time. So it's pretty interesting that you walk into a room with DNA and you come out with a virus. It doesn't quite happen that fast, but it is pretty quick. And then just to try to convince you that I'm not making all of this stuff up, so this is the structure of an adenovirus, and people figure these out using very, very advanced equipment where they're shooting protons and electrons and things at it, and they figured out that this is the shape of an adenovirus, and it's a pretty interesting structure. It looks like a honeycomb, and then it has these fibers sticking out of the top, and much like the coronavirus that uses the spike, the adenovirus uses these fibers to attach to a host cell and infect it. So that's what it was predicted to look like. Other people have taken electron micrograph images of it, and that's what it looks like. And here's the one that we made. So we can't quite see the fibers as well. If you have a high-res picture, I think there's one there, I think there's one there, and a couple around, but the shape looks right. So I'm pretty sure that our team at the Menzies Institute for Medical Research has made an adenovirus. It's not for COVID, it's for Tasmanian devils. But it's fairly easy to do. And now that you have the process down, we could probably make one of these in 3 weeks not 3 weeks, 3 months with the right funding and the right team. Places like AstraZeneca and Merck that have billions of dollars to throw at it, they can cut that timeline down quite a bit. They could probably have a first batch of it in a month and have it scaled up in somewhere from 3 to 6 months. The vaccines will not be able to keep up with the changes that we're seeing in the virus, though, because it's not just about making a vaccine, it's about testing it then, right? So nobody wants a vaccine that's not effective and not safe. So even if you could make a vaccine in a few weeks, which you probably could with those mRNA vaccines, you can't test them quick enough. So this is the idea of we're probably not going to reach herd immunity through a vaccine because the variants are getting ahead of the vaccines. But the vaccines will remain very useful in the community at keeping the number of viruses down. The more virus out there, the more variants you're going to get. So the take-home messages from this are that the vaccines are highly effective, especially initially, but as the variants are coming around, efficacy is going to drop. Vaccines can be made quickly, but probably not as fast as new variants can be made. And the technology that's coming out of these new vaccines is really going to drive forward some other positive things in biomedical research in the coming years. So a small silver lining here. Thank you.
Thanks very much, Andy. That was fantastic. So we've got part 2 of the immunology team with Guna here. He's going to talk some more vaccine.
Thank you very much, everyone. Good evening. So my background is I'm an immunologist and also a virologist. And I was also trained in epidemiology, so I would like to try and get some of the things I wanted to get to you quickly. So as a virologist, of course, it's something that I really show my students. But RNA viruses, which our SARS coronavirus 2 is one of them. And the thing about these viruses is that the genome is RNA. And another property of RNA viruses is that because the genome is RNA and they need to replicate, they tend to make lots of mistakes. And that's really the reason why we get variants. Okay? They are not like DNA viruses, which can actually fix mistakes because the DNA viruses depend on DNA replication of cells. Okay? And the other virus I've put up here is influenza. That's another virus which makes a lot of mistakes during replication, and those mutations can result in viruses that can be more infectious, more lethal. I also need to say something about transmission. As a virologist, what interests us is, viruses can get transmitted in many different ways. It could either be through contaminated food. It could be through intravenous injection with drugs, and then that's a way of getting infected with hepatitis B or C viruses. But the thing about these respiratory viruses is that they can be transmitted so easily and rapidly because we all need to breathe. We need to live. Okay, and I think most of us agree that the original virus, the SARS coronavirus 2, probably came from a bat, and then there was some intermediate animal, and then it got transmitted to us. Okay? But there have to be some really important changes for a virus from a bat to get into humans and cause disease. And that's what I want to talk to you next, and that is what Andy has alluded to. A virus with its spike proteins has to bind to receptors that are on our cells in order to gain entry into the cell. Only then can a virus replicate and cause disease. Right? And the receptor is called ACE2, and really one of the key functions of ACE2 is to regulate blood pressure. It also regulates wound healing and inflammation. And so it is a really important molecule, and the virus actually uses that as a receptor, and a lot of things go wrong. And what this whole idea of vaccinating against the spike proteins is that we want to block the proteins, the spike proteins present on the virus, from binding to our cells and that's going to stop anyone getting infected. Okay, so the immune system, obviously when we get exposed to a virus, we get infected, there is a period, incubation period, that's what we call, and then there is an acute phase when people get really sick, and then there are three different possibilities that people actually completely recover, which is on the left-hand side, on the very left. With SARS-CoV-2, we know that people can get infected and be asymptomatic. Usually it's in children, but we do know now that children can get infected and they can actually transmit disease. The worst case scenario is someone actually dies from the infection, and we know that there's been lots and lots of people who have died from SARS-CoV-2 infection already. And then, of course, the other outcome can be recovery, but people get symptomatic, so they get very sick, but they recover. But some people who recover do go on to develop what's known as long COVID. So they still have a lot of other effects from the infection. So all of that actually depends on our immune system, because the immune system, obviously, what I've got at the very top are many different cell types. Let's not worry about exactly what they are, but those cells at the very top are the ones that respond very rapidly to an infection. And if it's a virus that our body has not seen before, they're going to respond by throwing everything possible at it. And the lower bottom is where are the cells that are important for vaccines and the way our vaccines work. So at the very bottom, the B cells, the B lymphocytes, are the ones that make antibodies. Okay? And that happens much later compared to the early cell types that jump in and try and contain the infection. Right? So the immune system does two things in this case. One is, obviously, it aims to protect the host, but in doing so, especially if it's a virus that's new, and we know this from influenza pandemics as well. So viruses that we've never seen before suddenly emerge, and a good classic example is the 1918 Spanish flu. So early response, late response, the two things that the immune system does is, one, to try and protect, but in the process, it can actually cause a lot of damage, and that's what happens with SARS coronavirus 2. Okay, and I'll just very quickly show you, these are not human lung sections, these are animal sections, but this is what happens. So, we breathe, we need oxygen, but what happens following this is an example of an influenza virus infected lung section, and within about 6 days, that's what you get. On the right-hand side is a lung which is filled with a lot of fluid and cells that infiltrate, and I'll just keep moving. As an immunologist, I needed to put this last one up, especially if we're talking about herd immunity. Okay, so this is from the RACGP. The thing is, whatever you might have heard about, whether there's going to be herd immunity or not, the most important message is every one of us, as many of us, need to get vaccinated. Right? And if you see at the very bottom here, Tasmania, if we all get vaccinated, that's in blue, there's no chance of any transmission in the state. And I'll stop there.
Thanks, Guna, for that whirlwind guide to immunology. Sorry to push on forward, but we've got lovely Nicola up next to talk about outbreak management.
Thank you. Okay, hi everyone, and thanks for inviting me along tonight. A slight change in focus. So I'm an infectious diseases epidemiologist, and although I work for UTAS now, most of my career has been spent as working in an applied epidemiology role in health departments across Australia. So as a result of that, like Nick referred to earlier, I was seconded across to work on the COVID response here in Tasmania. I have recently returned to my role. So I thought the topic of tonight's seminar was tantalising and wondered how I could respond to that. And after speaking to Nick, I thought that yes, I'll talk about the future of outbreak management and what that means from an applied sense, but also about, from an epidemiological point of view, how we're going to navigate to living with COVID-19 from here. So others, my colleagues have spoken about herd immunity, but basically COVID-19 is not like, for example, smallpox. So if imported, it won't spread. COVID's not like that. And we know, and my colleagues have been talking about the vaccine, it's not a transmission blocking vaccine. So these things are all going to impact on herd immunity. The vaccine rollout and distribution is uneven, and I'm not just talking about the stoush going on at the moment between jurisdictions in Australia. Of course, internationally, the vaccine rollout is very uneven, and that's going to have a big impact on whether we can have anything anywhere near herd immunity. Of course, there's individual immunity variability that my colleagues have been talking about, and also the new variants. I think it's going to be, when we're talking about when we're going to see the end of this pandemic, I think it's going to be quite some time before we just think of COVID-19 as an endemic disease. We're very much still in a pandemic phase, and we won't be able to say that we're in that post-peak phase until most countries have dropped below their peak observed levels. And we really don't know when that will be for many countries. We've seen that pandemics are characterised by waves, and we're really not sure for a lot of countries whether we've seen the highest waves. When we do get to that post-pandemic time, the anticipated level of COVID disease activity will really vary across the world and locally as well, and there'll be pockets where it's very different, even in a place like Tasmania. There could be seasonal differences, and certainly, like I said, many differences by location. So the end of the pandemic isn't close. We know that there's vertical takeoff of cases in countries without or with low vaccination. So vaccination is absolutely critical. I think you're hearing that from us all. But alone, it's not a magic bullet. So we have got the strategies that you've all been hearing about, and those strategies have to change and vary very much dependent on how much vaccine coverage you have, 60, 70, 80, 90%. And our lives with COVID-19 will be different over the coming years. We, of course, in Tasmania virtually achieved elimination. We've called it suppression, but virtually we've had elimination in this state. We haven't seen a community-acquired case in over a year. So for us here, it's a very big step towards having circulating virus. I would say that we need to be very cautious around relaxing our protective measures. And you've all probably heard now the TTIQ. Public health responses include, of course, test, trace, isolate and quarantine. That will continue, but just in a slightly different moderated version. We're still going to have case management. We're still going to have some quarantine of contacts. And of course, we are going to have to respond to localised outbreaks, and particularly when they're occurring in risk settings. We will continue with some density caps for a while. And we'll have some moderated border restrictions and moderated lockdowns. No one wants to hear that, I know, but we won't be just all at once suddenly saying there are no restrictions. There'll be localised ones. We might just need to have borders around particular areas. And you probably would all recall that early last year we had to have a restriction around the northwest of Tasmania when we did have our outbreaks here in Tasmania. So there'll be those types of things happening as we move forward. We'll have to continue with our behavioural changes that we've been learning over the last 18 months or so, including social distancing when we can, hand hygiene, wearing of masks, which we haven't done a lot of in Tasmania, but we will be, and perhaps using the QR codes and certainly getting tested when sick. And the level of response, like I said, and ongoing restrictions will be very much influenced by what vaccine coverage we have. And we do need to meet those very aspirational targets in some groups, such as our healthcare workers that we've been hearing a lot about in the media in the last week or two. We know that our vaccinated individuals are very much less likely to get infected, and it's the best way to prevent infection. And we also know that individuals that have had COVID-19 should still get vaccinated because they're twice as likely to get reinfected if they don't get vaccinated. We also know that vaccinated individuals who do get infected experience a lot less symptoms, much more minor symptoms, and sometimes no symptoms at all, which is really the gold star, isn't it? As an individual, that's what you want. If you're going to get infected, you hope that you don't get serious disease. It also will help, of course, and very importantly, in managing rates of hospitalisation and serious illness at a population level, which is really important. But of course, the flip side of that is the workplace implications, particularly for those working with high-risk populations, if they're vaccinated and they perhaps don't have symptoms but have the ability to transmit disease. So it's not a straight line, and it's very complex. One size won't fit all, nor is it going to be static. It will be moving all of the time. As we've heard, models have limitations and assumptions. We'll continue to have pockets of low coverage, and we'll have hard to reach communities, and not just geographically, communities that aren't wanting to be vaccinated, for example. And we'll continue to have those people in our population with underlying health conditions or other risk profiles that we need to protect. And we very much have to keep an eye on the local health capacity. It's very complex, as we've been hearing, and changes rapidly, and we're continually improving as we go, and that has an impact as well. There's outbreak management plans that we're all trying to follow, but we change them constantly. And our level of response will have to change, and our type of response will have to evolve as well. Any significant relaxation at the moment is likely to result in a rapid rise in cases and hospitalisation, and we've witnessed that around the world in countries that have had a reasonable level of vaccination and have relaxed everything, and you've seen that sharp rise in cases. And we'll need to use these social restrictions to flatten the curve. And you'll all recall at the beginning of the pandemic, everyone suddenly knew what a curve was, what we were talking about, and everyone was interested in flattening it. But we've lost that language somehow more recently, and we're counting cases and wanting to know who they're linked to. But we will need to go back to that kind of concept of saying we need to have the curve at a state so that we can reduce the level of disease to a manageable level so that our hospitals aren't overrun, for example. And we'll use the lowest level of restrictions possible to ease that burden on the community whilst keeping that curve at a level that's manageable. And we really will have to monitor that strain on the public health and hospitals and intervene where we need to. So another thing probably to keep in mind too is that outbreak management isn't new. So for someone like me that's worked for decades in outbreak management in health departments, it's kind of our business as usual, and we do have really established, reliable methods that we're counting on for a lot of what we do. And there's a lot of this sort of public health action that you see happening with COVID. It all is slightly different per disease, but you can see there's a lot of other notifiable communicable diseases that we take action for. And I just thought I'd give you a couple of examples. So measles, for example, is a notifiable condition. We consider it very urgent when we get a measles case notified. It's vaccine preventable, it's highly contagious, and we don't have endemic spread here in Australia. And if we have a case, we treat it like an outbreak. That case is isolated, and we investigate it to identify the sources of infection, identify other cases, and protect any susceptible contacts. So you can see there's a lot of similarities to what we do in our usual business. TB is another one, another notifiable condition. The public health priority, how we treat that really is dependent on how infectious the case is. And infectious cases are isolated to minimise, again, ongoing transmission, and then we will investigate and identify their source of infection as well in other cases and protect susceptible contacts. Unlike measles, which is highly infectious, and COVID's infectious as well, but not as infectious as measles, TB is not as infectious, and we take a different approach to that, whereby we'll follow up contacts who are the closest and have spent, say, at least 8 hours with that case, and see whether we've got any disease in those, and if we have, and we might go out to the next circle and follow up there. And we might see ourselves doing a similar thing with COVID as time goes on, and we might use genomics to help us with that. And Louise is going to talk to us a bit more about genomics in a moment. And we might do, rather than massive follow-up that we're seeing now, where we want to find every person that's been in contact with every case, we might do a more focused way, where we go, there's a case in a class, we'll follow up that class and see how that goes, and will we then need to go out further in the school? We might not do this same level of follow-up that we've been doing up until now. We certainly, like I said earlier, will have to keep an eye on what's happening and give a heads up to health services, let them know if there's increased circulation of disease in their area, and protect them as much as possible. And sometimes when we follow up outbreaks as well, of course, it can lead to outcomes like increased vaccination as you follow up people. Monitoring of notification data, so the data you're seeing coming in for COVID and the vaccine uptake and things, we do that routinely across other vaccine preventable diseases as well, and use that data to inform our vaccine programs. And we'll be doing the same thing, and we are doing the same thing at the moment with COVID. And we'll be looking at vaccination type and at genomic strains and comparing them and taking public health action accordingly. So I think I've almost talked long enough. So to close, I suppose I wanted to say this pandemic is ongoing. Unfortunately, it's not over. It's going to be quite some time before we can say that it's an endemic disease and we're out of the pandemic. I think we need to be really cautious about relaxing our protective measures and do it step by step. We have to continue with those public health responses. We need to maintain and normalise our behaviour changes. We need to vaccinate, and then monitor and address outbreaks as they occur. Thanks very much.
Thanks very much, Nicola, for that grounding and always hard truths. It's my pleasure to introduce Dr. Louise Cooley. He's going to be our last speaker this evening. She's going to talk more about the Tasmanian situation and some genomics as well. So thank you very much.
Get it up.
Your keyboard.
Keyboard, right, thank you.
This one.
Perfect, okay. All right, thanks Nick, and thanks for inviting me to talk. It's been a great session tonight. So I'm just going to talk a little bit about the role of the laboratory in managing the pandemic. Not so much on how the pandemic ends, because although it will end, COVID will probably be around for a long, long time. The focus really, I'll talk about test, trace, isolate and quarantine, and how the laboratory plays a supplementary role to the public health measures. A little bit about how we do it, so upscaling our test capacity, implementing genomics, some local application of genomics and the future. So we began the pandemic sort of back in January 2020. We promised 150 tests a day to the government, using the first rule of always under promise and over deliver. And you can see on this, we sort of about a month ago, we were sitting at about 2,000 tests a day, so it's been a huge growth for us. We're lucky in some ways. We've got a very experienced molecular laboratory. We had a pandemic back in 2009, just trying to remember. So swine flu gave us a lot of lessons in how to set up high-scale testing and how to set up clinics in the community. So we were able to apply some of these lessons. We had one high-throughput testing platform. At this time, we had a single Tasmanian health service, which has been incredibly useful doing the large bulk testing that we're doing. We've got good working relationships with public health, so Nicola, and as well as, really importantly, we've had good relationships with our suppliers. And sitting behind all of this was a national laboratory network, which I'm the state representative, so the Public Health Laboratory Network, which really swung into action as soon as the COVID-2 emerged. on the bad side. We had insufficient stockpiles. We had international closures. We ordered our reagents and Chinese New Year hit. Our lab was too small. We were single supplier dependent. Transport networks weren't in place. And I guess most importantly is the uncertainty that happens at the start of a pandemic. How deadly is it? Is it going to come to Tasmania? Of course it's not going to come to Tasmania. You know, all the uncertainties that you deal with at the beginning, and yet you have to continue to make decisions in this sort of vacuum. I've stolen the Economist picture because I really liked it. So a lot of innovations taken place in the laboratories. We've now run almost 300,000 tests and our daily capacity is heading up towards 5,000 a day. Despite all of this, we've kept our turnaround times to less than 24 hours. So if you have a test in Burnie, you do have to wait 10 hours longer for your result than if you have your test in Hobart. But this is really vital for public health to get these results through quickly, because we know that in some states it can be up to 7 days, and if you're reporting cases that were tested 7 days ago, that's just not, you're not going to be able to control your outbreak. A lot of IT work, so our key person in the laboratory was actually our IT guy who helped with integration of the booking systems, the clinics, the laboratory. We interfaced everything, so essentially there's very little manual data entry. We used as you can see, these little racks up here. So at the start of the pandemic, everything was coming in single plastic bags. We got rid of those, put them into trays, and that's markedly reduced our waste and helped us just do this sort of industrial level of testing. And our other innovation, I guess, is, yes, I've just put wastewater testing as one of the other things we've taken up. So on to trace. A diagnostic test is useful. You get a binary result, detected, not detected. Almost all the time you're right. But that is really the beginning of the story of public health investigations. So to do more precise outbreak management, you need that increased granularity, and the way that we can get that increased granularity is by sequencing the SARS-CoV-2 genome of that particular case. And at the time that SARS emerged, there was a newly developed national microbial genomics framework that Tasmania had been involved in, as had the rest of the public health laboratories in Australia. And there were some baby steps just taking place there. So it really was a good time to work further on this genomics framework. So essentially the framework's about facilitating consistent and standardised genomic sequencing, the analysis and reporting in Australia. A platform had already been developed, which was called AusTracker, and this was selected to be the national platform. It acts as a central repository to everybody feeds their genomic data into it. We use it to detect and monitor variants of concern, and most importantly support national and international, because New Zealand are included in this, surveillance for public health units to track cases. I'm not going to go through this, but this just shows how comprehensively the framework sits under the auspices of AHPPC, which you may hear in the media, which is the principal health protection committee in Australia. And just to the right just shows all the laboratories that are involved in it. So in operation, you have a case, you get a sequence, and in general the average sequence time is between 1 to 2 days across Australia. The sequence is then uploaded to AusTracker. AusTracker team then, if they're happy that it passes the quality metrics, because it needs to cover about 60 to 70% of the genome, it's analysed, it's entered in, and a tree is made, which I'll show you in a moment, which puts your sequence in context of all the other sequences nationally. Every day the tree's uploaded, we can have a look at it and see where our particular isolate sits. If we see it sitting in a cluster, in particular one that we didn't expect, we'll let our public health unit know, and they contact the public health unit in that particular state, or multiple states as it may be, and do some further investigation. And to date, the sequencing rates per case range between about 40 to 92%. Tasmania are only having just over 230 cases of top Australia in the percentage of cases sequenced. 43 are the states that are just absolutely floundering under thousands of cases per day, but they usually go back and will do that backlog. So this is what it looks like, and this is a real screenshot from AusTracker. And in the circle there is a group of 4 identical sequences, and these were for some international hotel quarantine cases. Nothing too surprising there. They all belonged to the same family. The isolate was identical, but it's always sort of reassuring when there's no surprises. This is a more recent case. All that black line is the New South Wales outbreak. And if you've had a look at trees before, you can see that it's sort of starting to break down into little tiny trees, which happens as an outbreak evolves. Right down the bottom where the red line there is this. So this is our Tasmanian isolate, and it's just sitting very nicely in that New South Wales isolate, in that New South Wales group. So again, probably no surprises, but no surprises are good. If it had turned out to be sitting somewhere completely different on the tree, then perhaps Tasmania would have had a different response to this case. So I think that sort of really gives people some reassurance that there aren't sort of stray, unexpected cases in Tasmania. The other thing that AusTracker does is just monitor and reports on the variants of concern. I have taken off the identity of this variant of concern, but I guess most people who've done a little bit of genomics will know. So this is actually our Delta strain. And each colour is a different state. So it just gives us a visual on tracking what's happening. Not much at all there from Tasmania, and I think that was before our case. And again, this is another picture of straight out of AusTracker. We report fortnightly, so this is just one moment in time. It looks at the different variants of concern, and they're coloured according to states. So again, for a public health unit, this is a really important thing to be able to see and to see where everything fits. Just briefly, I've got enough time. Just quickly go through the Tasmanian ones. Okay, so this is our outbreak curve. Now, Nicola may not agree with me here, but this essentially we get a result. We get a sort of a black and white picture of yes, there's cases there, and this is what genomics can do. So it really adds in some colour and some detail and much more information about what's happening. So some surprises were found in looking at our Tasmanian isolates. So the northwest outbreak, which we had presumed was one, was actually 2 different isolates. So that 2 different outbreaks. Sorry. So that's the orange and the red. A few separate sort of ungrouped ones, and some small clusters that we were aware of because they'd been on a boat together or travelling together. One thing that this did find, though, were 3 separate, what we had called unlinked cases, spread over a number of weeks. So they're there with the purple arrows, and then you can see if you look up at the top of the firework, those cases sit together. And these were actually all involved, I guess, loosely in the tourism industry. Some were travellers, some were in the service industry of travellers. And these cases, when we went into AusTracker, actually linked very closely with cases from South Australia, Queensland, New South Wales. So we've never really had time to get to the bottom of this, because I think following this, the public health units have been so busy. But that's something that we do want to do one day, is go back and have a look at these cases and actually see, because these were all, it's actually a small undetected outbreak within Tasmania. Okay, so post-pandemic world, test, trace, isolate, quarantine will be a part of our life for some time yet. How intensive that will be will probably just depend on how much virus is around at the time, and also, of course, new variants. So as a new variant comes in, the intensity will probably increase. The laboratory will continue to play a key role in diagnostic, surveillance and public health management. Variants will continue to emerge, and probably the most recent one is C1.2, which looks like a very nasty emerging strain, which has mainly been detected in South Africa. Some of these will become a dominant strain, some will completely disappear, and it's totally unpredictable. So only time will tell that. And the community response to the pandemic, as a microbiologist and a clinician, we've really seen some change in the viral dynamics and viral epidemiology during the pandemic. So, for example, RSV, so respiratory syncytial virus, we did not see it all last year. We had a very early season. This season it was a very bad season. So lots of newborns, 1 and 2 year olds getting RSV for the first time and now quite unwell. And then of course influenza, which we have not seen since March last year. So final thoughts. What of influenza? So we have been lucky, but influenza will come back. So and we as a laboratory are preparing for that time when we have influenza and SARS-CoV-2 circulating. And finally, keep away from Facebook. I'm not on Facebook, but one of the comments that we saw on Facebook were when we had 11 cases in the Northwest, one comment was, well, are they only testing 11 specimens a day? So a friend of the laboratories very kindly made us up some mugs that said proudly testing as many as 11 samples per day. So keep away from it. Okay, and there's just all my acknowledgements. Thanks, Nick.
Thanks so much, Louise, and thank you all to the fantastic speakers. Big round of applause for all our speakers tonight. So we have gone over time quite a bit, but we have definitely time for questions. So I hope you have some. So passing it on to the floor. Is there any questions? Out the back, yes.
My apologies for the ignorance of what follows. My academic discipline is philosophy, not any of those. It's related to the general question of variants and vaccines chasing variants. And 2 comments. One, can someone speak to the idea of having one vaccine for the first dose followed by another vaccine for the second dose and why this might be a good idea? So that's question 1. Question 2 is, on the idea of vaccines, I understood that most of them targeted proteins on the tip of the spike. And the trouble was that a lot of the variation in vaccines occurred in those tip proteins. So you had a vaccine that targeted one particular, the tip changes, well, of course, it's no longer applicable. You've got to tweak the vaccine. I understood that there was a French mob whose name escapes me, whose vaccine targeted the stem of the protein, which wasn't changing so much. So it sounded like it might be more variant-proof than some of the other things that are rattling around the ridges, all of which might be ignorant and false, but I'd be pleased to have comment.
Okay, team immunology, perhaps? That seems like a question for both of you.
So both very good questions. Is the microphone coming through? Yeah. So both very good questions, and I think the idea of combining vaccines is probably a good one. The major issue with it is just making sure that it's safe. So the AstraZeneca vaccine has actually been combined with some of the Russian vaccine, the Gamaleya one. They're both adenoviral vectors, but one of the issues with using the same vector over and over is that your body gets immune to the vector, and then it can't deliver the SARS-CoV-2 spike protein as well. So combining those would work well, and there is clinical trials with those 2 in combination. I think out of necessity, some of the other ones are being trialed together, and time will tell how well they're working together. So I think that could work. It's just a matter of making sure things are safe first. As far as targeting the tip of the spike protein, I think you're on target there, that most of the vaccines will encode the whole spike protein. The tip is the key area because that's where it interacts with the receptor that Guna was talking about. And there probably are some that maybe are targeting the stem a bit more. What I would actually look for a bit more, and there are some in the clinical trials, is putting different viral proteins in also. So there's a nucleocapsid, which the RNA wraps around, and that one is much less prone to evolution and mutations. It's a more fixed structure. The problem with that one, though, is it's inside the virus. So when you make an antibody, it can't get that. But once the virus gets in, it starts making copies of itself. There's other cells in the body, like T cells, that can come by and see that because it gets chopped up inside the cell and then put to the surface, but it can't block the transmission. And that's what the antibodies are good at blocking, is transmission, and that's what we want.
I just want to
Yeah, I just want to follow up on what Andy was saying about the mixing of the vaccines. So there's a lot of evidence, at least not with the SARS-CoV-2 vaccines, but so we know from a lot of other studies, at least in animal models, that combining different types of vaccine platforms induces much better immunity. So if we take AstraZeneca, for example, it is a recombinant virus. And the reason why we have to wait for about 3 months is if we do the vaccination in a short period of space, our immune system is going to make antibodies to the adenovirus vector as well as the spike protein as expressed. And so if we do it too soon, the second shot is not going to be effective. So if you come along with, say, Pfizer as the second vaccine dose, that seems to actually work. There is some evidence that it actually works better. And we also know from other vaccines against other diseases as well.
Right, great. We've got a related question actually online, and the question is, could you please explain why the vaccines can stop infection but don't stop transmission? I think that's the question. So maybe to the immunologists as well. So how can they not stop transmission but stop being infected?
Don't lose it.
Oh.
And that works.
Excellent.
You don't need to, you can just talk.
Press on?
No, it's green. It's fine.
Okay.
Please.
So vaccines they will reduce your chance of getting infection, but they don't prevent infection in all people. So once you've been you need to be infected before you can transmit. So I think that's it, essentially, that you can still get infected, so you can still transmit. It's just your chances of getting infected are lower, significantly lower. And of course, your chances of having severe illness or death following vaccine are extremely low. So about 90% reduced.
Yep, fantastic. Hope that answers your question. Any other questions from the audience? Yes, down the front here. Oh, yep, there, sorry, that's fine. Yep, you're first. Second.
Sorry, I'm a second-year medical student, so I should really know this. But Guna, I think you said in one of your lectures, is it the neutralizing antibody response that we're actually looking for in the vaccine rather than the T-cell response?
So if I understood your question correctly, you are asking whether it's the neutralizing antibody rather than the T-cell mediated immunity? So if we are using a live viral vaccine, so in the case of smallpox, that's going to induce both antibody responses and T-cell mediated immunity. With these vaccines, the mRNA vaccine and the AstraZeneca, they're just going to induce antibody responses. That's really what we want. But the best vaccines, of course, would be something that would induce both cell-mediated immunity and antibody responses.
Yep.
Does that answer your question?
Yes, thanks. Also, the Doherty Institute, I think they said that there's the risk of tens of thousands of cases, even in the best case scenario. As a medical student next year in the hospital, I don't really want to be, in an environment where, our teaching is sort of once again shifted online, like a likely possibility.
I guess that's the issue that public health and particularly Mark Veitch will be wrestling with at the moment. If you step right back to the vaccination story, a lot of the aim of vaccination is not only protecting ourselves and protecting the other people in the community, but it's protecting our health system as well. So when we're a totally naive population, we know that a significant proportion of people, be it 0.1% up to 2%, depending on which demographic you're looking at, will end up in hospital, and the hospital systems will become overwhelmed, and that's when you don't get your teaching. If we have high vaccination rates, we are likely to still see cases. We are likely to still see some severe cases, and anybody who isn't vaccinated at all will still remain very susceptible to severe disease. But overall, we anticipate our future will be continuing to see cases of COVID, but having that severe cases and the overwhelming of the health system, the aim will be to prevent that. And that's where the physical distancing, all the social measures come in. So if we start to see that, as Nicola said, the vertical rise, that really steep climb in numbers, then we'll be told to change our behaviour. And that might be very local, it might be whole of state. It just really sort of depends on each scenario.
Yep. Thanks for that.
Excellent. We had a question down in the front here. Wait for the mic, Cut's coming.
Thank you. My question is more of a general one regarding vaccines into the future for us. We talk about now having resistant to antibiotics across the population. Is there a future where we can see that we no longer be able to make vaccines that will be able to use the same vectors or address new viruses, because we've been using vaccines, the individual has so many in a lifetime, the world now is having it across the board. Is it something that we're going to basically use up the possibility of making vaccines for future? Does that make sense?
I'm happy to take the first go at that. So with the three most primarily used adenoviruses, one is a chimpanzee adenovirus, that's the AstraZeneca one.
Yeah.
The Johnson & Johnson is a human adenovirus serotype 26, and the Russian Gamaleya uses the serotype 26 and then a human adenovirus serotype 5. So in humans, there's at least 55 adenoviruses, so there's quite a few that could be used if we were running out of those vectors. We could go into chimpanzees, and that's a much smaller population than the humans, but there'd be a bunch there. So there is a rich trove of resources that we could get new viral vectors from if we needed. There's certainly other types of viruses that have been used as vaccines. So herpes viruses have been modified for cancer vaccines. So there's a lot of them out there that we can use. The mRNA ones are quite promising because it doesn't induce the same response against the vaccine. It's kind of a lipid-based structure on the outside, and it doesn't have a lot that the immune system can go after. It gets in the cell and then it does its work. So it's kind of an amorphous blob that gets into the cell.
So it's a finite resource, but it's not a one that we need to be concerned about for future.
Probably not.
situations.
I mean, the good thing, one of the few good things about COVID is that we've done these massive tests and we've seen that this chimpanzee one is relatively safe. There's some issues with clottings and things, and some of the other ones we know those are safe, but each time we want to use a new one, we're going to have to do big clinical trials to make sure they're safe before you go putting them in people.
Okay, and the other thing is, given the amount of vaccines that we have during an individual's lifetime, is that going to ever become an issue, that the vaccines, we carry them through our lifetime? So do they work against each other or cause issues, counterproductive to each other?
I'll have a go at it. So if you think about the fact that if we do not have vaccines, we are going to be infected with all kinds of pathogens. And so it comes back to the question, are we going to run out of something? So I don't think so.
So we are running out of time, alas. We have time for at least one more question from the audience, one from the online. Any others? Oh, anyone else? Oh, over here. Yes.
No,
right down there.
Someone else.
Yeah. Go over this one. This one, please. Yeah. Yeah. Oh, right. Yeah.
Watching the daily news conferences by the premiers has been fascinating, ranging from the grumpy Dan Andrews to the cool mom Gladys Berejiklian. And I was just looking at their ABC digital news at the moment. It says New South Wales promised bright summer, quotation marks, as planned to end COVID-19 lockdown revealed. That's from Gladys. What are the prospects for a bright summer? Seems pretty remote to me.
Take that one.
I mean, is this just a con job on?
I'll have a go. Like I was saying earlier, it's going to vary considerably, and there are differences of opinion on this. But I don't think even New South Wales is suggesting that it will be completely open. There will still be a level of restriction in place, and they certainly will. They've made it quite clear too that as they relax their restrictions, if things start to get further out of hand, that they will have to draw it back in. Even with their regional lifting of restrictions, I think it's tomorrow night, they have said that that will change should they see the situation changing. So, it's nice to remain positive about this, and hopefully we will have a bright summer, but it won't be a completely free summer even New South Wales.
Do you think it will be advised?
I think we're going to have a lot of cases still at Christmas time, but I think that we will have a level of vaccination that will allow us to have a lot more freedoms than we have at the moment.
Fantastic. Okay, so one last question was about AstraZeneca perhaps, and why the changing of the times between vaccinations. Would, again, the immunologists like to address that one? So reducing the time.
So Guna touched on this a little bit. So when you get the first AstraZeneca vaccine, your body is recognizing not only the spike protein that it's making, but also the virus itself. So I showed those pictures of the honeycomb-like structure, if anyone was paying attention and remembers the picture. But the immune system is going to recognize that honeycomb-like structure, and the antibodies are going to hit it before it can get inside the cell and then start making that spike protein, because the spike protein isn't there part of the virus. That's only made once it gets in the cell and starts trying to make copies of itself. So if you have this first dose, there's a lot of antibodies against adenovirus. It blocks from getting in the cell, and then you're not making the spike protein that you need. So by letting it go about 3 months, your body's making antibodies, it's going up, and then they start waning back down. So when you get the next dose of the AstraZeneca vaccine, more of those viral vectors can get inside the cell and make the spike protein, and you'll get a better secondary response by putting a bigger gap there.
Yeah. Oh, thanks for that. And thanks to all the speakers for a fantastic session. And thanks also to Erik Volz in London as well. He's still online. So thank you very much, Erik.